Hey, welcome to a Moop Knowledge, or a, what is this channel called now? To a Moop Math? I think it's Moop Math. So Moop Math is a channel about all academic subjects, trying to master all of them, to aid students, to offer tutoring services, to help you answer homework problems, and to act as kind of like a hub where people can help each other with homework or any kind of problem for people in school, people in high school, university, college, technical school, or even like self-learners at home, older people or younger people who are just interested in higher level stuff. So today I'm going to be reading some biology. So here we go. Here is some biology. So we left off last time with biology talking about microfilaments. So microfilaments are solid rods about 7 nanometers in diameter. They are also called actin filaments because they are built from molecules of actin, a globular protein. Microfilament is a twisted double chain of actin subunits. See table 7.2. So I actually can't show you the table. I'll try to avoid saying that. Microfilaments seem to be present in all eukaryotic cells. In contrast to the compression-resisting role of microtubules, the structural role of microfilaments in the cytoskeleton is to bear tension, pulling forces. So we're talking about the microstructure inside the cell right now. In combination with other proteins, they often form a three-dimensional network just inside the plasma membrane, helping support the cell's shape. This network gives the cortex, the outer cytoplasmic layer of a cell, the semi-solid consistency of a gel, in contrast with the more fluid state of the interior cytoplasm. In animal cells, specialized for transporting materials across the plasma membrane, bundles of microfilaments make up the core of microvilli, delicate projections that increase the cell surface area. So microfilaments are well known for their role in cell... Just move the microphone here. Are well known for their role in cell motility, particularly as part of the contractile apparatus of muscle cells. Thousands of actin filaments are arranged parallel to one another along the length of a muscle cell, interdigitated with thicker filaments made of a protein called myosin. Myosin acts as a motor molecule by means of projections, arms that walk along the actin filaments. Contraction of the muscle cell results from the actin and myosin filaments sliding past one another in this way, shortening the cell. In other kinds of cells, actin filaments are associated with myosin in miniature and less elaborate versions of arrangement in muscle cells. These actin-myosin aggregates are responsible for localized contractions of cells. For example, a contracting belt of microfilaments forms a cleavage furrow that pinches a dividing animal cell into two daughter cells. Localized contraction brought about by actin and myosin also play a role in amoeboid movement, in which a cell crawls along a surface by extending and following into cellular extensions called pseudopodia, from the Greek pseuds, false, and pod, foot. Pseudopodia extend and contract through the reversible assembly of actin subunits into microfilaments, and of microfilaments into networks that convert cytoplasm from sol to gel. Sol being the fluid state of cytoplasm. Not quite. There's a lot of fluid states that are not liquid or gas. There's a lot of different fluid states, and cytoplasm is one called sol. Okay, where were we? According to a widely accepted model, filaments near the cell's trailing end interact with myosin, causing contraction. Like squeezing on a toothpaste tube, this contraction forces the interior fluid into the pseudopodium, where the actin network has been weakened. The pseudopodium extends until the actin reassembles into a network. Not only do amoebas move by crawling, but so do many cells in the animal body, such as white blood cells. In plant cells, both actin-myosin interactions and sol-gel transformations brought about by actin may be involved in cytoplasmic streaming, a circular flow of cytoplasm within the cell. This movement, which is especially common in large plant cells, speeds the distribution of materials within the cell. I'm going to grab some water. 1 second, guys. Enjoy the techno. Or more specifically, enjoy the psychedelic trance. Okay, I'm back. Now we have, in plant cells, both actin-myosin interactions and sol-gel transformations brought about by actin may be involved in cytoplasmic streaming, a circular flow of cytoplasm within cells. This movement, which is especially common in large plant cells, speeds the distribution of materials within the cell. Intermediate filaments. Intermediate filaments are named for their diameter, which at 8 to 12 nanometers is larger than the diameter of a microfilament, but smaller than that of a microtubule. Specialized for bearing tension, like microtubules, intermediate filaments are a diverse class of cytoskeletal elements. Each type is constructed from a different molecular subunit belonging to a family of proteins called keratins. Microtubules and microfilaments, in contrast, are consistent in diameter and composition in all eukaryotic cells. Intermediate filaments are more permanent fixtures of cells than are microfilaments and microtubules, which are often disassembled and reassembled in various parts of a cell. Chemical treatments that remove microfilaments and microtubules from the cytoplasm leave a web of intermediate filaments that retains its original shape. Such experiments suggest that intermediate filaments are especially important in reinforcing the shape of a cell and fixing the position of certain organelles. For example, the nucleus commonly sits within a cage made of intermediate filaments, fixed in location by branches of the filaments that extend into the cytoplasm. Other intermediate filaments make up the nuclear lamina that line the interior of the nuclear envelope. In cases where the shape of the entire cell is correlated with function, intermediate filaments support that shape. For instance, the long extensions, axons of nerve cells that transmit impulses are strengthened by one class of intermediate filament. Various kinds of intermediate filaments may function as the framework of the entire cytoskeleton. Cell surfaces and junctions. Having crisscrossed the interior of the cell to explore various organelles, we complete our tour of the cell by returning to the surface of this microscopic world, where there are additional structures with important functions. Although the plasma membrane is usually regarded as the boundary of the living cell, most cells synthesize and secrete coats of one kind or another that are external to the plasma membrane. Plant cells are encased by cell walls. The cell wall is one of the features of plant cells that distinguishes them from animal cells. The wall protects the plant cell, maintains its shape, and prevents excessive uptake of water. On the level of the whole plant, the strong walls of specialized cells hold the plant up against the force of gravity. Prokaryotes, fungi, and some protists also have cell walls, but we will postpone discussion of them until Unit 5. They were currently in Unit 2. Plant cell walls are much thicker than the plasma membrane, ranging from 0.1 micrometers to several micrometers. The exact chemical composition of the wall varies from species to species and from one cell type to another in the same plant, but the basic design of the wall is inconsistent. Microfibrils made of the polysaccharide cellulose see figure 5.8, are embedded in a matrix of other polysaccharides and protein. This combination of materials, strong fibers in a ground substance matrix, is the same basic architectural design found in steel-reinforced concrete and in fiberglass. A young plant cell first secretes a relatively thin and flexible wall called the primary cell wall. Between primary walls of adjacent cells is the middle lamella, a thin layer rich in sticky polysaccharides called pectins. The middle lamella glues the cells together. Pectin is used as a thickening agent in jams and jellies. When the cell matures and stops growing, it strengthens its wall. Some plant cells do this simply by secreting hardening substances into the primary wall. Other cells add a secondary cell wall between the plasma membrane and the primary wall. The secondary wall, often deposited in several laminated layers, has a strong and durable matrix that affords the cell protection and support. Wood, for example, consists mainly of secondary walls. I'm going to grab some more water because I can barely speak. Came back. So the extracellular matrix, ECM, of animal cells functions in support, adhesion, movement, and regulation. Although animal cells lack walls akin to those of plant cells, they do have an elaborate extracellular matrix, ECM. The main ingredients of the ECM are glycoproteins secreted by the cells. Recall that glycoproteins are proteins with covalently bonded carbohydrate, usually short chains of sugars. The most abundant glycoprotein in the ECM of most animal cells is collagen, which forms strong fibers outside the cells. In fact, collagen accounts for almost half of the total protein in the human body. The collagen fibers are embedded in a network woven from proteoglycans, which are glycoproteins of another class. Proteoglycan molecules are especially rich in carbohydrate, up to 95%. They can form large complexes as shown in figure 7.9. Oh, my bad. Some cells are attached to the ECM by still other kinds of glycoproteins, most commonly fibronectins. Fibronectins bind to receptor proteins called integrins that are built into the plasma membrane. Integrins span the membrane and bind on their cytoplasmic side to microfilaments of the cytoskeleton. Thus, integrins are in a position to transmit charges in the ECM to the cytoskeleton, and vice versa, to integrate changes occurring outside and inside the cell. Current research on fibronectins and integrins is revealing the influential role of the extracellular matrix in the lives of cells. Communicating with the cell through integrins, the ECM can regulate a cell's behavior. For example, some cells in a developing embryo migrate along specific pathways by matching the orientation of their microfilament to the grain of fibers in the extracellular matrix. Researchers are also learning that the extracellular matrix around a cell can influence the activity of genes in the nucleus. Information about the ECM probably reaches the nucleus by a combination of mechanical and chemical signaling pathways. Mechanical signaling involves fibronectins, integrins, and the cytoskeleton. Changes in the cytoskeleton may in turn trigger chemical signaling pathways inside the cell. In this way, the extracellular matrix of a particular tissue could help coordinate the behavior of all the cells within that tissue. Direct connection between cells also function in this coordination, as we discuss next. Intercellular junctions help integrate cells into higher levels of structure and function. The many cells of an animal or plant are organized into tissues, organs, and organ systems. Neighboring cells often adhere, interact, and communicate through special patches of direct physical contact. It might seem that the non-living cell walls of plants would isolate cells from one another. In fact, as already mentioned, plant cell walls are perforated within channels called plasmodesmata. Singular would be plasmodesma, from the Greek word desmos, to bind. Cytosol passes through the plasmodesmata and connects the living contents of adjacent cells. This unifies most of the plant into one living continuum. The plasma membranes of adjacent cells are continuous through a plasmodesma. The membrane lines the channel. Water and small solutes can pass freely from cell to cell, and recent experiments have shown that in certain circumstances, particular proteins and RNA molecules can also do this. The macromolecules to be transported to neighboring cells seem to reach the plasmodesmata by moving along fibers of the cytoskeleton. In animals, there are three main types of intercellular junctions: tight junctions, demosomes, and gap junctions. All three types are especially common in epithelial tissue, which lines the internal surfaces of the body. Figure 7.30 uses epithelial cells, of the intestinal lining to illustrate the junctions. Each kind of junction has a structure well adapted for its function. At tight junctions, the membranes of neighboring cells are actually fused, forming continuous belts around the cells. These junctions prevent leakage of extracellular fluid across a layer of epithelial cells. In our example, the tight junctions of the intestinal epithelium keep the contents of the intestine separate from the body fluid on the opposite side of the epithelium. Desmosomes, also called anchoring junctions, function like rivets, fastening cells together into strong sheets. Intermediate filaments, made of the sturdy protein keratin, reinforce desmosomes. Gap junctions, also called communicating junctions, provide cytoplasmic channels between adjacent animal cells. Special membrane proteins surround each pore, which is wide enough for salt ions, sugars, amino acids, and other small molecules to pass. In the muscle tissue of the heart, the flow of ions through gap junctions coordinates the contractions of the cell. Gap junctions are especially common in animal embryos, in which chemical communication between cells is essential for development. The cell is a living unit greater than the sum of its parts. From our panoramic view of the cell's overall compartmental organization to our close-up inspection of each organelle's architecture, this tour of the cell has provided many opportunities to correlate structure with function. This would be a good time to review cell structure by returning to the various figures. But even as we dissect the cell, remember that none of its organelles works alone. As an example of cellular integration, consider the microscopic scene. The large cell is a microphage. It helps defend the body against infections by ingesting bacteria. The macrophage crawls along the surface and reaches out to the bacteria with thin pseudopodia, philopodia. Actin filaments interact with other elements of the cytoskeleton in these movements. After the macrophage engulfs the bacteria, they are destroyed by lysosomes. The elaborate Endomembrane System, which includes the Endoplasmic Reticulum and the Golgi apparatus, produces the lysosomes. The digestive enzymes of the lysosomes and the proteins of the cytoskeleton are all made on ribosomes. And the synthesis of these proteins is programmed by genetic messages dispatched from the DNA in the nucleus. All these processes require energy, which mitochondria supply in the form of ATP. Cellular functions arise from cellular order. The cell is a living unit greater than the sum of its parts. Okay, very cool. So now we're going to summarize some concepts. So, microscopes provide windows to the world of the cell, and improvements in microscopy have catalyzed progress in the study of cell structure. Cell biologists can isolate organelles to study their functions, and they use the ultracentrifuge to produce pellets enriched in specific organelles. Prokaryotic and eukaryotic cells differ in size and complexity. All cells are bounded by a plasma membrane. Bacteria and archaea have prokaryotic cells without nuclei or other membrane-enclosed organelles. All other organisms have eukaryotic cells with membrane-enclosed nuclei and other membranous organelles in their cytoplasm. The need for a high surface-to-volume ratio limits cell size. Internal membranes compartmentalize the functions of a eukaryotic cell. Plant and animal cells have most of the same organelles. The nucleus and ribosomes. So the nucleus contains a eukaryotic cell's genetic library. DNA is organized with proteins into thin fibers of chromatin, which coil to form thick chromosomes in dividing cells. Associated with the chromatin in non-dividing cells are one or more nucleoli, sites of ribosome synthesis. Macromolecules and ribosomal subunits pass between nucleus and cytoplasm through pores in the nuclear envelope. Ribosomes build the cell's proteins. Free ribosomes in the cytosol are and bound ribosomes on the outside of ER and the nuclear envelope synthesize proteins. The endomembrane system. Many of the eukaryotic cells' membranes are connected either by physical continuity or through transport vesicles made of pinched off pieces of membrane. The Endoplasmic Reticulum manufactures membranes and performs many other biosynthetic functions. Continuous with the nuclear envelope, the Endoplasmic Reticulum, ER, is a network of cisternae, membrane-enclosed compartments. Smooth endoplasmic reticulum lacks ribosomes. It synthesizes steroids, metabolizes carbohydrates, stores calcium in muscle, and detoxifies poisons in liver. Rough ER or endoplasmic reticulum has bound ribosomes and produces cell membrane and secretory proteins. These products are distributed by transport vesicles budded from the ER. The Golgi apparatus finishes, sorts, and ships cell products. Stacks of separate cisternae make up the Golgi. The cis face of a Golgi stack receives secretory proteins from the ER in transport vesicles. The proteins are modified, sorted, and released in transport vesicles from the transface. The Golgi also synthesizes some macromolecules on its own. Lysosomes are digestive compartments. Lysosomes are membranous sacs of hydrolytic enzymes. They break down cell macromolecules for recycling as well as substances ingested by phagocytosis. Vacuoles have diverse functions in cell maintenance. A plant cell's central vacuole functions in storage, waste disposal, cell growth, and protection. So other membranous organelles. Mitochondria and chloroplasts are the main energy transformers of cells. Mitochondria, the sites of cellular respiration in eukaryotes, have an outer membrane and an inner membrane that is folded into cystae. Some reactions of respiration occur in the mitochondrial matrix enclosed by the inner membrane, and others are catalyzed by enzymes built into the inner membrane. Chloroplasts contain chlorophyll and other pigments that function in photosynthesis. In chloroplasts, two membranes surround the fluid stroma, which contains thylakoids stacked into grana. Okay then. Peroxisomes generate and degrade H2O2 in performing various metabolic functions. Peroxisomes carry out processes that produce hydrogen peroxide, H2O2, as waste, and their enzymes convert the toxic peroxide back into water, H2O. And also releases oxygen. Providing structural support to the cell, the cytoskeleton also functions in cell motility and regulation. The cytoskeleton is made of microtubules, microfilaments, and intermediate filaments. Microtubules grow out from the centrosome, a region near the nucleus that includes two centrioles in most animal cells. Microtubules shape the cell, guide movement of organelles, and help separate the chromosome copies in dividing cells. Cilia and flagella are motile appendages containing microtubule doublets that are moved past each other or by the motor protein dynein. Microfilaments are thin rods built from actin. They function in muscle contraction, amoeboid movement, cytoplasmic streaming, and support for microvilli. Intermediate filaments support cell shape and fix organelles in place. Cell surfaces and junctions. Plant cells are encased by cell walls. Plant cell walls are composed of cellulose fibers embedded in other polysaccharides and protein. The extracellular matrix, ECM, of animal cells functions in support, adhesion, movement, and regulation. Animal cells secrete glycoproteins that form the ECM. Important components include collagen, proteoglycan complexes, and fibronectin attached to integrins in the plasma membrane. Intercellular junctions help integrate cells into higher levels of structure and function. Plants have plasmodesmata, channels that pass through adjoining cell walls. Animal cell contact is by tight junctions, desmosomes, and gap junctions. The cell is a living unit greater than the sum of its parts. So now we have some quiz questions. The symptoms of certain inherited disorder in humans include respiratory problems and in males sterility. Which of the following is a reasonable hypothesis for the molecular basis of this disorder? So a defective enzyme in the mitochondria, a defective actin molecules in cellular microfilaments, defective dynein molecules in cilia and flagella, abnormal hydrolytic enzymes in the lysosomes, and a defective secretory protein. So respiratory problems would mean there's something wrong with the microphilia in the lungs that catalyze the oxygen going into the blood, red blood cells, and carbon dioxide being removed. And for the sterility, it'd be a lack of whatever produces steroids, which I believe is lysosomes. So I think it would be D, abnormal hydrolytic enzymes in the lysosomes. And let's see if we have the answer. And that is actually incorrect. It is defective dynein molecules in cilia and flagella. Okay, so we can review that later. Choose a statement that correctly characterizes bound ribosomes. Bound ribosomes are enclosed in their own membrane. Bound ribosomes are structurally different from free ribosomes. Bound ribosomes generally synthesize membrane proteins and secretory proteins. The most common location for bound ribosomes is the cytoplasmic surface of the plasma membrane. Bound ribosomes are concentrated in the cisternal space of rough endoplasmic reticulum. So the answer for this would be C. Bound ribosomes generally synthesize membrane proteins and secretory proteins. So that's what they do. Okay, number 3. Which of the following organelles is least closely associated with the endomembrane system? A, the nuclear envelope, B, the chloroplast, C, the Golgi apparatus, D, the plasma membrane, or E, the endoplasmic reticulum. So this would be the B, the chloroplast. So 4. Cells of the pancreas will incorporate radioactively labeled amino acids into proteins. This tagging of newly synthesized proteins enables a researcher to track the location of these proteins in a cell. In this case, we are tracking an enzyme that is eventually secreted by pancreatic cells. Which of the following is the most likely pathway for movement of this protein in the cell? Endoplasmic reticulum to the Golgi to the nucleus, Golgi to the ER to the lysosome, nucleus to the ER to the Golgi, ER to the Golgi to the vesicles that fuse with plasma membrane, or ER to the lysosomes to the vesicles that fuse with plasma membrane. So the answer to this is, of course, D. It goes from the endoplasmic reticulum to the Golgi to the vesicles that fuse with the plasma membrane. Okay, that's all the time we have for today for bio. And yeah, I hope you guys enjoyed the hard rave music in the background and the psychedelic visuals. And I will see you guys next time. Goodbye.